In this video, we will briefly go over how the CRISPR-Cas9 method works. The CRISPR system is a bacterial immune system that allows bacteria to protect themselves against viruses. These viruses, known as the bacteriophage, first bind to the bacterial cell and injects its DNA inside of the cell. The virus then uses the reproductive machinery in the bacteria to assemble and reproduce. The virus will continue to reproduce inside of the cell until the bacteria cell bursts and, quite frankly, dies. In this armed race that's been going on for billions of years between viruses and bacteria, viruses seem to be unstoppable, killing trillions of bacteria every single day. When all hope seemed to be lost, the bacteria developed a virus-killing immune system that would change the way we edit genes forever. Now, let's get into how this mechanism actually works. The CRISPR-Cas9 defense mechanism works in 3 stages. The first stage is the viral DNA acquisition stage. In the bacterial DNA, there is a segment of DNA that contains repeated sequences that are interspaced by spacers. These spacers contain the DNA of invading viral DNA from prior infections. When a virus infects a cell, bacterial protein cas9 and cas2 acquire a segment from the DNA known as the protospacer and is added to the CRISPR array as a new spacer. This process is similar to a photographer taking a photo of the viral DNA and storing it onto a hard drive. It serves as a memory storage unit for all prior invading viral DNA. So to summarize this stage, if the bacteria survives the virus, it will store the viral DNA in an archive known as CRISPR. Stage 2. The CRISPR array is then transcribed into a pre-CRISPR RNA. The single-stranded RNA contains a segment of all prior viral DNA and the repeated sequences. Another RNA, known as the tracer RNA or transacting RNA, binds to the pre-CRISPR RNA and forms a double RNA duplex. This strand of DNA is then cleaved to a manageable size, forming now a mature CRISPR RNA, which is what they call guide RNA. The Cas9 protein, which is going to be the protein that's going to cut the gene, binds with the guide RNA and now can easily find its target. To simplify this stage, the storage unit, as mentioned previously, makes a copy of the entire file containing the photos of the DNA from all the previous invading viral DNA. This file is trimmed into a photocopy and given to a bacterial bounty hunter so that he knows who he needs to kill. Simplifying this step even further, the bacteria is making an RNA copy and arms the Cas9 protein. Stage 3. If the guide RNA matches the viral DNA, then the Cas9 protein will bind to it, unwind it, and cut the DNA sequence. But wait, recall that the bacteria stores the viral DNA in its own DNA sequence. The viral DNA that is targeted by the Cas9 is the same DNA that is in the CRISPR array. So how does the Cas9 protein tell the difference between its own DNA and the enemy DNA? The answer is the PAM. The PAM, which stands for protospacer adjacent motif, is a specific sequence of nucleotides, about 2 to 6 nucleotides long, that is directly next to the protospacer, hence the name protospacer adjacent. This specific sequence, GG, must be present for a Cas9 to bind with the viral DNA, serving as a recognition site for the Cas9. So how does this PAM sequence prevent Cas9 from binding to its own DNA? The spacer sequence in the CRISPR array are not followed by GG. The sequences of the repeats are always the same. This prevents the Cas9 from binding to its own DNA because it lacks the recognition site GG. So how is there always a PAM sequence at a true Cas9 targeting site? In the DNA acquisition stage we discussed previously, we mentioned that the Cas1 and Cas2 protein acquire the photospacer. When doing so, they only choose sequences that directly follow a PAM sequence. Only picking sequences with PAMs guarantees that when the same viruses invade again and the Cas9 is armed with its RNA guide, nothing will stop it from destroying the enemy DNA. Here you will see the Cas9 protein containing the guide RNA. The Cas9 protein will recognize the PAM sequence, bind to the DNA molecule, and 2 tiny molecular scissors will cut the DNA. Humans use this system by allowing it to basically try and repair itself. When it does so, this process is error-prone, causing mutations that will disable the gene. Another way we use this is once the gene is cut, we replace that gene by introducing a good copy of the gene, tricking the cell to think our strand of DNA is its own. The cell will try and repair itself using our DNA strand we introduced. Well, there you have it. That pretty much summarizes how the CRISPR-Cas9 method works.